Welcome to Anatomy and Physiology at Glen Oaks Community College. I'm Dr. Ren Hartung. For this video, I'm going to talk about the bare bones basics of membrane potentials. So what is membrane potential? This drawing is supposed to represent a cell, a human cell. Here's the cell membrane, the outermost part of the cell, and that's where the action is when we're talking about membrane potentials. And there, of course, is the nucleus, just to show you that this is a cell. When we're talking about membrane potentials, here's the basics of what we mean. There's a charge across this outside membrane, a literal electrical charge. It's a little bit more positive on the outside and a little bit more negative on the inside. By the way, when these are reported, like in your textbook, for example, we're measuring from the inside. So resting membrane potential is almost always reported as a negative number because we're measuring relative to what's happening on the inside. To understand the basics of how this kind of charge difference works, a good first step is to make sure you understand how the cell membrane is made up. So let's take a quick look at the structure of the cell membrane. If I took a little piece of the cell membrane and blew it up a lot, this is what we think we would see, these little structures. And by the way, let me grab one of them and draw it large or larger. These are called phospholipids. They have a phosphate head group, and that phosphate head group likes water. It's attracted to water. Water easily associates with it. It's hydrophilic, in other words. That's the term for that, hydrophilic. Whereas the tails down here, they don't like water. They're made out of lipids. And lipids don't have polarity or charge. So they actually don't like water. They tend to stay away from water. That's the phospholipid, and those are the characteristics that matter in terms of why they associate the way that they do in cell membranes. So let's get back to the membrane structure. Phosphate head group, lipid tails, and if I kept drawing this thousands and thousands of times, you could imagine that I could go all around my cell membrane here. That's what this is made out of. But of course, we don't draw it that way because doing that would take forever. What this creates in terms of a barrier, in terms of a semi-permeable barrier, these phospholipids, in the way they associate with each other, it's called a phospholipid bilayer, that creates a lipid barrier. These tails hanging out with each other creates a lipid barrier so that molecules that are polar or charged, particles that are polar or charged, they can't get through this barrier easily. Proteins are required in the membrane to allow things like this to pass through. So for example, the two major ions for membrane potential, sodium and potassium, this cell membrane, if it was only made out of phospholipids, these guys wouldn't be able to get through. They'd just bounce right off and they'd stay either outside the cell or inside of the cell. They wouldn't be able to pass across this membrane. Again, understand the structure of the cell membrane and in what way it's a barrier is the beginnings to understanding membrane potential. Let's move on from that. You understand the cell membrane. The next step to go to is, remember I said that in order for these guys to get through, there need to be proteins. The two major proteins for setting up membrane potential. The first one is called the sodium-potassium pump. It literally pumps sodium and potassium across the membrane. In order to do that, it requires energy. And of course, the energy currency inside of the cell is ATP. This protein is going to take that ATP and break off one of the phosphate groups, turning it from adenosine triphosphate into adenosine diphosphate, or ADP. And then, of course, we'll have that phosphate group, which, by the way, is negatively charged. The energy from removing that phosphate group is used to do this, to pump out 3 sodiums, and at the same time to pump in 2 potassiums. To pump out 3 sodium ions and to pump in 2 potassium ions. This creates a difference in concentration, of course, of sodium on the outside of the cell and potassium on the inside of the cell. There's more potassium on the inside of the cell, less potassium on the outside, and there's more sodium on the outside of the cell, less sodium on the inside. It's important to get that concentration difference, connect that with your understanding of diffusion, because when we talk about how these potentials can change, that comes in real handy. Just in terms of charge, though, because remember what we're trying to do here is explain how it is that the cell is positive on the outside, negative on the inside. Part of this is explained here, at least a little bit, just in terms of our accounting of charges. Notice that this sodium-potassium pump has pumped out 3 positive charges. And while it has pumped in positive charges as well, it's only pumped in 2 positive charges. So we already have more positive charge on the outside, less positive charge on the inside. We've got a piece of the negativity going on here. In other words, the inside of the cell, just talking about the sodium-potassium pump, is a little bit more negative because it has fewer positive charges. Let's go to the next protein. This next protein is called the potassium leak channel, and it does exactly what its name says. It literally allows potassium to leak out. Remember, that's what we were pumping in over here. So the sodium-potassium pump pumped out some positive charge. It pumped out more positive charge than it pumped in. And now those positive charges that the sodium-potassium pump pumped in, we're allowing some of those to get out, thus increasing even more the positive charge on the outside and decreasing the positive charge or increasing the negative charges on the inside. These 2 proteins that I've just described, those are the 2 major proteins for setting up resting membrane potential. They set it up and they maintain it. And again, that's why the cell is positive on the outside and negative on the inside. And that is the bare bones basics of membrane potential. Other things to mention, there are other ions that influence membrane potential. Calcium ions can influence membrane potential, for example. Chloride ions can influence membrane potential. Basically, any charged ion can influence membrane potential. Calcium ions are important in terms of the membrane potential changes that cardiac myocytes or heart cells undergo. Chloride ions with their negative charge, they're important for some of the membrane potential changes that occur in neurons. So while sodium and potassium ions are the major ones involved with membrane potentials, I don't want you to think they're the only ones. These aren't the only ones either. Any charged ion can have an influence on it, potentially at least. But because I don't want to risk going too far and giving you too much information right now, that's the bare bones basics of membrane potentials. So quick quiz to make sure you are paying somewhat attention to this. Let me erase your helpers here. Just at the bare bones basics, what are we talking about when we say membrane potential? Of course, the answer to that is we're talking about the difference in charge across a cell membrane. And for most cells, that charge is a little bit positive on the outside and a little more negative on the inside. What are the 2 proteins, the 2 major proteins involved with setting up membrane potential? And the answer to that is the sodium-potassium pump and the potassium leak channel. Let's continue with those 2 channels. What does the sodium-potassium pump do, and how does it do it? And the answer to that is a little bit more involved, but it's still pretty simple. To be honest, if you can draw this same picture that I drew earlier, ATP being broken down into ADP plus P, and then that energy from the ATP being used to pump out 3 sodium ions and to pump in 2 potassium ions. If you can reproduce this picture for yourself, That will answer almost any question that you might get about the sodium-potassium pump. And I'm assuming you're not in graduate school and learning about its protein structure and all these things, but just bare bones basics for understanding the sodium-potassium pump. And this is a great one to know because it comes up over and over again. It's used a lot in living cells. So that's the sodium-potassium pump. Next question. What's the other protein involved with resting membrane potential or with setting up membrane potentials? And the answer to that, again, we already did that one, but it's the potassium leak channel. So what does it do? What does the potassium leak channel do? And the answer to that is, I drew it over here. And again, you can just remember it simply as a drawing. It allows potassium to leak out of the cell. And by the way, if you're ever confused on which direction the potassium is going to move, remember that this is simply a channel. It's just open. So the potassium is going to move according to diffusion. And remember, the sodium-potassium pump was pumping potassium in, so there's going to be a higher concentration of potassium in here. Therefore, the potassium is going to diffuse out of the cell from an area where potassium is at higher concentration to an area where potassium is at a lower concentration. By the way, for those of you who are learning anatomy and physiology now, you may have had biology classes in the past. And you might be getting bells rung in your head because you're like, oh, I think we did this in biology a little bit. These kinds of things, understanding diffusion, understanding the cell membrane, understanding proteins in the cell membrane and how they basically function, to me, those are like the basics of understanding human biology in general or anatomy and physiology in general. We could even go back to chemistry in terms of understanding what we mean when we say hydrophobic and hydrophilic, and what kinds of things like water and what kinds of things don't water. So anyway, those basics should always be there. You should practice them and memorize them because they'll come in handy even later on when you're helping with patients and things like that if you're going into the medical field. All right, I'll get off my high horse. Thank you once again for watching, and as always, if you have any questions, please feel free to leave them in the comments down below.